So hello everyone, my name is Mohamed Magassa, and I would like to thank my lab members. I would like to thank the Neurological Surgery Summer Student Program, and I would like to thank the Neurological Surgery Department. Here are my funding acknowledgements. And so, brief background on intracranial aneurysms. An intracranial aneurysm is the outpouching of a diseased blood vessel within the brain, and a ruptured aneurysm can cause a hemorrhagic stroke, which may result in significant morbidity and mortality. So approximately 2 to 3% of the population will develop an intracranial aneurysm, and roughly 30,000 aneurysms will rupture on a yearly basis within the United States of America. So aneurysms are fatal in about 40% of cases, and out of everyone who survives, about 66% will suffer permanent or some type of neurological deficit. So here are some symptoms and some contributing factors to aneurysm formation, growth, and rupture. So when it comes to traditional treatments of intracranial aneurysms, what a surgeon will usually go for is a surgical clipping in which they will perform a craniotomy, removing part of the skull, and depending on the location, the size, and the shape of the aneurysm, they will choose to clip it. And fast forward to modern treatments of intracranial aneurysms, a surgeon will usually go for an endovascular coil and stents, in which a coil would just fill the inside of the aneurysm dome. And a stents, there are two types of stents. This particular one is preventing the coil from herniating back into the parent vessel. And so the next stent is called a flow diverting stent, in which it has a higher metal surface area, which acts as a scaffolding for the parent vessel to heal. So if you take a look at this image on my left, you'll notice two blood vessels within the brain. So this here is the ICA, this is the ECA, this here is the MCA, and this here is the ACA. So endothelial cells line the inside of the arteries, and these specific endothelial cells at this location here are experiencing the force of the blood as it tries to go through this bifurcation. So that is causing these endothelial cells to dysfunction of forming a cerebral aneurysm. So our hypotheses: one, there are specific endothelial markers of vascular dysfunction, and differential hemodynamic stress are dysregulated during aneurysm formation. And two, the level of gene expression is dependent on the aneurysm's risk profile as defined by the phases aneurysm risk score. So phases is a clinical scale which is used to predict the risk of rupture for intracranial aneurysms, and it takes into consideration 6 variables: patient population, patient age, whether the patient had hypertension, whether the patient had a previous hemorrhagic stroke, aneurysm size, and aneurysm location. So our aims are to collect endothelial cells to perform targeted transcription measurements and to compare the differential profile of key vascular factors. So when it comes to our methods, of course, the first thing that we will have to do is gain the patient consent in order to biopsy the cells from the devices which will be put into their blood vessels during surgery. And these devices are usually thrown out, and it could be such as coils and catheters. So the way that process would work is that we will stretch the device out, rinse it with PBS in order to gain all of our cells. And then after we move on to cell counting, in which we will have to centrifuge the cells, get them down to a pellet, and then we would add a buffer in order to lyse the red blood cells, because the only cells that we're really looking for are endothelial cells. And so we will then stain cells, which will allow us to count them, and then we will move on to flow cytometry. And the way that flow cytometry works is that every endothelial cell has a specific cell receptor, and so we will want to tag a fluorescent marker to an antibody, which will then bind to the cell receptor of the endothelial cell. And so with that process, we'll be able to gain our endothelial cells in one group and then another pile of just anything else. So we will put these cells inside of the machine, and the machine will be able to separate the cells based on the type of fluorescent marker which is tagged to it. And then we move on to RNA sequencing, and then which we look at mRNA in order to tell protein's abundance and its function. So from our results, we just started in July. However, we were able to gain biopsy devices from a total of 3 patients so far. And the way that this process went is that we'll be in the angio suite, as we talked of earlier, and the surgeon will clip the coil into a tube. We will then transport the tube in ice over to a collaborator's lab over at South Lake Union. And then after we get there, we will spin the cells down, plate the cells, and we will allow it to culture for a total of about 5 days. And then we will look at the cells under a microscope. So if you look at this image here, So these images are from patient number 2, and we let the cells culture for about 5 days. And these cells here are red blood cells, which culture. And this image here, we're hoping to have some endothelial cells within some of these clumps. So some lessons that we learned from Dr. Cook are, one, the diameter of coils. So the larger the diameter, the more of a chance that it will have to scrape endothelial cells from the inside of the dome. First coil, so the first coil always seems to pick up a lot more endothelial cells rather than the second or the third. And natural cell yield, so it's normal to gain a low amount of endothelial cells, but that just means that we have to be extra careful with our procedure. So we know that there is limited data concerning the genetic expression of endothelial cells, and that is because of risks associated with surgical tissue collection of intracranial aneurysms and arteries. And we also know that fewer and fewer patients are undergoing surgical clippings, in which we would like to expand our research to the growing endovascular population. And so Dr. Cook was actually able to perform the endovascular biopsy on a total of 10 patients. He looked at 5 ruptured aneurysms and 5 non-ruptured aneurysms. And he was able to find a total of 437 endothelial cells, and out of these cells, 94 appeared to be aneurysmal cells. So the next evolution of our project, we would like to combine what Nee spoke of earlier, the model seeding project, where she has patient-specific flow and anatomy, and we would like to combine it with my endovascular biopsy, in which I have patient-specific cells. So we'll be able to have an in virtual model of an aneurysm with patient-specific endothelial cells, patient-specific anatomy, and patient-specific flow. And we're hoping to find something interesting with that. So I would like to thank Dr. and Mrs. Ellenbogen for making this program a possibility. We are definitely a group of 13 students who are lucky, and we appreciate everything that you've done for us. I would like to thank Julie and Sylvia for all the help that we've received throughout this program and coordinating. I would like to thank the whole lab, easily the best lab that I've ever been at, and I would like to thank Dr. Kim and Dr. Levitt for everything that I've learned. And thank you.